Okay, so let's see. First of all, information presented is not an official opinion of Michigan State University or the University of New England. So that's me, Zach Burton, and Lele is my co-host, and we're completely responsible or irresponsible for all content presented. So this is Life on Earth, Origin of Life webcast, and the topic is the origin of life on Earth. So a broad topic and a topic in which we're going to try to push some ideas about integrating various information and putting it together to come up with an understanding of, or a partial understanding of the origin of life. So Lele, that's Lei, at University of New England, Biddeford, Maine, in the School of Biological Sciences. And this is me. I was a professor at Michigan State University for 30 years in the Department of Biochemistry and Molecular Biology. So MSU is where the recent school shooting occurred, sadly. Okay, so what are we talking about? So these are the subliminal messages. I think what I'm presenting is the greatest story in about 4 billion years. I'm going to try to push the idea of integration of transfer RNA evolution, evolution of tRNAomes. A tRNAome is all the tRNAs of an organism. Aminoacyl tRNA synthetase evolution. So those are the enzymes that put an amino acid on a tRNA, and also genetic code evolution. So it's a lot to put together. And here, mostly what we're trying to do is to give you an idea of what you need to integrate in order to make sense of what it is we're trying to present. So there are bottom-up and top-down approaches to origin of life. A bottom-up approach would be to make life in a test tube, which remains a challenge, but perhaps is becoming an easier challenge. And a top-down approach, that's to infer, in this case, origin of life from existing biology, which is tRNA sequences. So the origin of life on Earth, and embedded in genetic code is a recorded history from about 4 billion years ago. That's because some of this history was recorded 4 billion years ago and never overwritten, and in particular that's seen in tRNA sequences. We'll present the 331 nucleotide mini-helix theorem. This is a theorem and not a model or a hypothesis because it's a proven hypothesis. Everything about this is completely correct with only a few minor ambiguities, which I'll point out. So aminoacyl tRNA synthetase enzymes, those are the enzymes that put an amino acid on a tRNA. Those are very important. And if you look at the pattern by which these radiated, it gives you the pattern by which the genetic code evolved, remarkably. Transfer RNA, I'm going to say, is a collectible, practically an antique at an age of about 4 billion years. Okay, so everything on this slide you can be absolutely certain of. The only ambiguities are in the red segments. So this loop segment here, that's a stem, that's a loop, that's a stem. These two loops, these loops. There's just a small amount of ambiguity in the sequences in those positions. So what am I going to tell you? So I'm going to tell you that there was once a polymer world that had regular polymers, And this is, everything on this slide is pre-life. Everything on this slide is chemical evolution. None of this is Darwinian evolution in organisms. So in polymer world, among other things, there were GCG repeats, CGC repeats, UAGCC repeats, and stem loop stems with these stem sequences and something very close to that loop sequence. And there was also a primitive adapter for attaching an amino acid. That was an ACCA sequence. And from polymer world, you get mini helix world. So in mini helix world, among other things, there are 31 nucleotide mini helices. So each of these sequences is 31 nucleotides in length. And this is a GCG repeat, a UAGCC repeat, a CGC repeat. This is a complement of that. And so this can form a stem loop stem, and this can form a stem loop stem where that's the loop. This is 7 nucleotides long. That's 7. 7 plus 7 is 14, plus 17 is 31. And these are stems of 5 and a loop of 7. So that's 17, and that's 17. This sequence and this sequence are identical. This sequence becomes the D loop of transfer RNAs. This sequence here becomes the anticodon loop, stem loop stem. That's a stem, that's a loop, that's a stem. And this one becomes the T loop, stem loop stem of tRNAs. So if you take this sequence and you ligate it there and you ligate this one there, you get molecules such as this one. And this is a 93 nucleotide precursor to transfer RNAs. Okay, this is a molecule that can be processed back into mini helices. So if you take 3 times 31, that's 93. This can be much longer. If you process it here, here, there, and there, you've replicated mini helices. And if you make this deletion here, a 9 nucleotide deletion inside a yellow and a green segment, the yellow segment is a 3' acceptor stem. The green segment is a 5' acceptor stem. And that's 7, and that's 7, that's 14. You delete 9, and you're left with 5, GGCGG, and GGCGG there. This is a type 1 tRNA, that's a type 2 tRNA. And these are primordial sequences. So these are sequences in pre-life. Over on this side, if you do nothing, you end up with a 3' acceptor stem ligated to a 5' acceptor stem. That's 7 plus 7, that's 14. That's the primordial length of a type 2 variable loop. The only difference of a type 1 tRNA is the length of the variable loop, and in this case it's 5, and it's those 5, and it's processed very similarly to this. So that's 7, and that's 7, that's 14. You delete 9, you're left with 5, CCGCC, 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 like that. So this is a fragment of a 3' acceptor stem, and this is a fragment of a 5' acceptor stem. So there are really 4 points that I want to make about this theorem. The first point is that this is based on a 100% ordered sequence. That is to say 100% RNA repeats and inverted repeats. So these are all, this is a repeat, GCG, that's a CGC repeat, that's a UAGCC repeat, that's a stem loop stem, that's an inverted repeat. And this is the only Slightly different feature, ACCA is a primitive adapter. You can attach glycine to that, and you can anneal that to a GCG repeat and bring two of these close together in an RNA environment, and you can make polyglycine polypeptides. A big purpose of polymer world, mini helix world, and initially of tRNA world is to make polyglycine. So this is a system to make polyglycine. This is a system when you add an ACCA to the 3' end of 31 nucleotide mini helices, you can bring two glycines close together in an RNA environment, and you can make polyglycine. And this system is initially to synthesize polyglycine, but this system here, tRNA, can train itself. It can learn through evolution to encode proteins, and that's how protein synthesis evolved on this planet. So the first point, 100% ordered sequence. Second point, anticodon 17 nucleotide stem loop stem, and the T 17 nucleotide stem loop stem are homologs. So let's look at that here. Stem loop stem for anticodon, stem loop stem for T. This sequence and that sequence are still closely related, essentially like nearly identical twins in this molecule. And this stem loop stem and that stem loop stem are also clearly related genetically. Initially, they were identical. The only ambiguity is what this particular loop sequence might have been. The stem sequences are known with certainty, as are all of the repeat sequences in mini helix world as we've drawn it here. The third point is that there are 3 mini helices, and you can count them up various ways. You can count them 1, 2, 3 like that. You can count them by mini helix cores, 1, 2, 3. Notice that this one is a different flavor than that one, and that one, that one, and that one were initially identical, and they've slightly deviated from one another over 4 billion years of Darwinian selection by customizing the loops for their various purposes. And then the fourth point is I've completely described type 2 and type 1 primordial tRNA sequences in this slide. If you want to go forward to this molecule, you make this internal deletion here, the same deletion there, and on this side, you make no deletion to make type 2 tRNA, and you make this deletion, which is related to that deletion, to make type 1 tRNA. And everything on this slide is certain. The only slight ambiguity is in the sequences of these 2 loops before LUCA, the last universal common cellular ancestor. So type 1 tRNA, that's a type 1 tRNA. Let's see. Okay, so this is a type 1 tRNA. You can tell that because it has this short yellow segment here. That's the variable loop, and that's 5 nucleotides. So if you don't believe me, that's 1, 2, 3, 4, 5 nucleotides connecting this cornflower blue segment to this cyan segment here. So that's 5, and that corresponds to these 5 right here. That's 5 out of 7 total for a 3' acceptor stem. So this is a fragment of a 3' acceptor stem. And this is included in my image, but hopefully it'll go up. Yeah, okay. So this is these 5 here correspond to those 5 here. That's 5 out of 7 total. That's a 5' acceptor stem. This is a fragment of a 5' acceptor stem in green. This is based on a CGC repeat, complementary GCG repeat, GCG repeat. This is a UAGCC repeat, where the D loop meets the T loop. That's called the elbow, and it looks kind of like an elbow on an arm. This is a Watson-Crick pair between a G and a C. And here you have a G intercalating between these two bases and forming a hydrogen bond to this U before the U-turn in the loop. Here you have the anticodon stem loop stem. Here you have the T stem loop stem, like that. Stems of 5. So 5 plus 5 is 10, plus 7 is 17. Same thing here. Here's a U-turn after a U in a 7-mer loop. Here's a 7-mer loop, a U-turn after a U. A U-turn is a U-shaped turn in the RNA backbone, as you can see there. A little bit less good view here, but this is a nice tight loop. And this is a tight loop to present a 3-nucleotide anticodon in purple. So this first position would be the wobble position. This is a Watson-Crick and a Watson-Crick position that interacts with messenger RNA. So messenger RNA codon goes 1 versus 3 in the anticodon, and 2 versus 2 in the anticodon, and then a wobble position in the third position. So 3 nucleotides, this one, that one, and that one, specify a single amino acid there. That's why tRNA is the genetic adapter for translation of proteins. Oh, at the 3' end, you have an ACCA. A is the initial discriminator sequence, which then diversifies in tRNAs of living organisms. 2 C's to interact with the ribosome and hold, in this case, a phenylalanine next to a growing polypeptide chain, and then you transfer the chain to the phenylalanine, and then you move this tRNA from the A site to the P site and bring in another amino acid attached to a tRNA into the A site on the ribosome to synthesize proteins. Got all that? Me neither, but we'll work with it. Okay, so that's our type 1 tRNA there. This is a typical tRNA from a Pyrococcus species. And this is a typical tRNA glycine from Pyrococcus. So typical means this is a consensus, in this case, of 46 tRNAs. That's all the tRNAs in this organism. If you're looking at Pyrococcus, you're effectively looking at LUCA, the last universal common cellular ancestor. So this is a living fossil of LUCA. LUCA can be considered one of the first organisms on Earth. So this is our primordial sequence here, as I just showed you in the 331 nucleotide mini-helix theorem. You can tell it's type 1 because it has a 5 nucleotide variable loop. So the sequence CCG, CC, CCG, CC, like that. You have a stem loop stem, another stem loop stem. This becomes the D loop. That's a UAGCC repeat. This is a GCG repeat. That's a fragment of a GCG repeat of a 5' acceptor stem. Anyway, just to show you that the model's correct, you have for the T loop sequence CCGGG, and here you have the sequence CCGG, often G, 7-mer loop, U-turn after a U, This is T in DNA, that's U in RNA. 7-mer loop, U turn after a U. These 17 and these 17 initially were identical. And now they're nearly identical after 4 billion years of Darwinian selection in tRNA in living organisms. And this interval here is also 17. And this sequence is UAGCC, UAGCC, switch over, U, G for A, G, C, C, UA, almost a perfect UAGCC repeat as in the model. And this is GCG, GCG, G, a perfect GCG repeat as in the model. And this used to be GGCGG as in GGCGG. It's slightly changed to improve the stem. And this used to be CCGCC as in CCGCC there, and it's only slightly changed. So basically, the model is very good. Primordial tRNA, a consensus typical tRNA, that's the same sequence, and a glycine tRNA in this organism is the same sequence. So that indicates that the primordial tRNA was an ancestral glycine tRNA. Glycine tRNA is closest to tRNA pre of all the Pyrococcus tRNAs, and all of the tRNAs in this organism radiated from tRNA gly. So type 2 tRNA, so that's a type 2 tRNA, and the only difference between this molecule and type 1 tRNA is this funny chicken wing here. And let's just go back briefly here. This yellow segment is 5 long. And that would be 1, 2, 3, 4, 5. And then it would link this up to the cyan position and delete 2 bases here in yellow and 7 here in green. 7 plus 2 is 9. So that's a 9 nucleotide internal deletion in creating a type 1 tRNA. Okay, and so these 7 here correspond to these 7 here. This is based on a CGC repeat. This is based on a CGC repeat. These 7 here correspond to these 7 here. This is based on a GCG repeat. This is based on a GCG repeat. This forms a stem loop stem. And I'll explain that in a little bit more detail in a bit. The discriminator, this is from Pyrococcus. This is an ancient tRNA leucine, practically a LUCA tRNA leucine. And this is an A, so that's the original ACCA, is the original amino acid adapter. And this is the discriminator base, that's initially A. Everything else I've shown you before. This is a UAGCC repeat. There's the elbow. That loop relates to that loop, that stem to that stem, that stem to that stem. And this sequence here relates to those 5 and those 5, like that. This is a U-turn after a U. That's a U-turn after a U. The loop is a little unwound, so this isn't the form of the loop during translation. During translation, it would be a tight loop, like this loop. That's one of the virtues of this loop, is it's a very tight loop to present 3 nucleotides as an anticodon. Okay, so this is type 2 primordial tRNA, and initially The V loop is a 3' acceptor stem. That's a 3' acceptor stem ligated to a 5' acceptor stem, like that. So 7 plus 7 is 14. In tRNA for leucine, it's 7 plus 7 for 14. The sequence is slightly changed. I'll explain why. And in tRNA serine, it's 7 plus 7 plus 1. There's the one base insert into the V loop in this particular organism, Pyrococcus. So this is a variable loop, and that's a variable loop. I've already explained all this stuff because that's the same as type 1 tRNA. So it doesn't really need to be explained again. This is a stem loop stem. There's an enzyme called LURS that puts leucine here and does not put leucine there. CRRS puts serine there, does not put serine there. So how does that work? So you have this stem, loop, and stem. LURS interacts with this A and G on the loop. The length is 14, as indicated down there. And LURS has to interact with these two bases to put leucine there. But LURS can't interact with two U's on this loop, and it can't reach those two U's because the attitude of this stem loop stem is different from the attitude of this stem loop stem. So there are two ways of looking at that. One way is for the angle of the hydrogen bonds. So these hydrogen bonds have a different angle than those hydrogen bonds. That's a Watson-Crick GC pair there. So you have the angle of these bonds, or you can score it this way. You have two unpaired bases between the 3' stem and what's called the Leavitt base pair there. So this G folds on that C. One of them flips over, so it's a reverse Watson-Crick base pair. And the same thing here, the Leavitt base pair named after Michael Leavitt. Here the score is 1, and a score of 1 is universal for archaea for tRNA serine, and a score of 2 is universal for tRNA leucine in archaea. And that scoring gives a different angle of this arm than this arm. LURS interacts with those two bases to put leucine there. LURS can't reach these two bases because of the attitude of this stem. And CRRS lies across the stem and interacts with the T loop. With this arm, the arm is too close to the T loop, and CRRS can't make that connection. So that's a little bit about how you get biological specificity into this system. Oh, this is a reminder. So UAGCC, UAGCC, two perfect repeats. In case you don't believe me that this is a UAGCC repeat, you should believe me, but you don't, you're under no obligation. And then if you switch back here, U, G for A, G, C, C, U, A. This is a G and not an A because it intercalates in there and interacts with that U. So it's selected to be G, and it's always G. And let's see, U, G for A, G, C, C, U, A, almost a perfect U, A, G, C, C repeat. The model's really very good. Okay, so the next thing I want to emphasize is the importance of the 7-nucleotide anticodon loop. So this is really specialized chemistry and important chemistry to understand if you care about origin of life. So there it is, and it's these 7 nucleotides here. It's a tight loop in the conformation that forms on the ribosome. And there's this interaction up here that's called a Hoogsteen pair. between an A and a C. And then there's a U, there's a U-turn, there's a wobble base, a Watson-Crick base, a Watson-Crick base, a heavily modified base, and that's the whole loop. So this loop is specifically formed to present three nucleotides as an anticodon to messenger RNA. So messenger RNA, the codon, you have a first codon base here, second codon base there, a third and wobble codon base here. So at a wobble position, you only have two possibilities, purine versus pyrimidine. You don't have four base discrimination. At a Watson-Crick position, Watson-Crick, Watson-Crick wobble, at a Watson-Crick position, you have four possibilities, AGCU and AGCU. But part of the reading of this base is this heavily modified base. So this heavily modified base maintains the frame of three nucleotides and doesn't allow a four nucleotide anticodon. For one thing, it can't form base pairs because of the modifications. So this is part of the reading of this base. And the other thing that makes this base a Watson-Crick position is a change in conformation of the ribosome that locks the mRNA and the tRNA together so that you enforce Watson-Crick geometry here and there, but allow and regulate wobbling at that position there. So this position here, I'm going to say, used to be a wobble position, and this used to be a wobble position. If you have two wobble positions, you have two possibilities here and four there, two there and four there. That's a complexity of 8 amino acids, so you hang up at 8 amino acids, evolving the code. When you lock this down by this modification and by the conformational change in the ribosome, now you have four possibilities there, four there, and two there. So 4 times 4 times 2, that's 32. So you expand from 8 amino acids potentially to, I don't know how many amino acids, fewer than 32. But in fact, we have 20 amino acids plus stops. So we got cheated out of a few amino acids. That has to do with degeneracy and genetic coding. So the genetic code evolved around the tRNA anticodon, these three nucleotides here. Initially, there were two wobble positions, that's 36 and 34. Both of these were wobbles. And then you suppress wobbling here, so you evolved to one wobble position here. Suppression of wobbling there, that involves 30S ribosome conformational closing, which enforces Watson-Crick geometry there. And then also heavy modification of the 37 base, which is part of the reading of the 36 base. So the genetic code, so this is somewhat complicated, but this is the genetic code in archaea, which are the oldest organisms, the most similar to Luca. So the ones we want to understand in terms of the evolution of life. And this is a codon-anticodon table. So the codon first position, that's messenger RNA, codon. Second position, third position. Third position is a wobble position, so there's no discrimination between pyrimidines or purines. And I'll partly explain that in a minute. This is the anticodon, so that's the tRNA. These two bases are the 34 position. This is the 35 position, and this is the 36 position here. So that's the 35 position. In column 4, it's always C. Column 3, it's always U. Column 2, it's always G for 35. Column 1, it's always A for 35. And the 36th position, that's rows. So row 1, 2, 3, and 4. Row 4 is the best row. That's because it's 36C all the way across. Okay. The next best row is row 2. That's 36G all the way across. And then row 3, 36U. And finally, and lastly, row 1, which is 36A. And the reason that row 1 is hard to fill is that when this is a wobble position, when that's a wobble position, essentially these tRNAs would act as stops. Let's see. And then the A and B rows, that relates to the 34 position. So A is never used in archaea in the wobble position. It's not utilized. And for the most part, it's not utilized throughout biology, except when it's modified to various things that change the way it functions. But G can be utilized because G can recognize U and messenger RNA as a wobble pair, or G can recognize C as a Watson-Crick pair. And U can recognize both A and G. G is a wobble, A is Watson-Crick. C mostly interacts with G, but U and C are mostly modified in ancient organisms. And particularly U is almost universally modified. And basically, you never separate these into two different amino acids because there's sufficient ambiguity with wobble C in tRNA or wobble U. Okay, so this is the anticodon, and this is the aminoacyl tRNA synthetase, the enzyme that charges the tRNA with amino acid. This is the amino acid. You can see these are all hydrophobic amino acids, and these are all 1A aminoacyl tRNA synthetases, so related enzymes for charging and related amino acids, and that's all in column 1. So that's lots of evolution in columns. That's evolution around the 35 base. And in column 2, same thing. These are all 2A enzymes. Serine and threonine are closely related amino acids. So that's lots of evolution in column 2. I'm going to say that alanine used to be a 2A enzyme, and then before Luca changed to a 2D enzyme to prevent translation errors. So there used to be more evolution in columns than you see today. Here you have orange, green, orange, green, orange, green. That's kind of an interesting pattern. You have lots of amino acids encoded in column 3. This is related to that, is related to that. This amino acid is closely related to that one. Here you have a 2B, a 2B, and a 2A enzyme. I'm going to say that this used to be a 2A enzyme and evolved to a 2B enzyme to suppress translation errors, and then this enzyme evolved from that enzyme. Here you have arginine and cysteine, which are both 1A enzymes. I know they've been classified other ways by other people, but this is a better classification, as I'll explain. So lots of evolution in columns. That's the 35 base right there. Code appears to fill by rows. Row 4 is favored because that's 36C. And then row 2, and then row 3, and lastly row 1 after you suppress wobbling at the 36 position. you can fill row 1. And columns 1, 2, and 4, 1, 2, and 4 have similarities to one another. And column 3, the most innovated, has this interesting stripe pattern that needs to be explained. That can be explained in a very straightforward way. So columns 1, 2, and 4 primarily sectored on the 35 and the 36 position. Column 3 primarily sectored on A and B rows, and that relates to the wobble position. So here and there, that's the A and B rows. So they sector, this is a B row, B row, B row, that's an A row, A row, an A row. Aspen, acin are related, glu and glin are related. And there's no A in a wobble position. So why this isn't written into every paper about translation, I have no idea, but I suggest that it become part of the lore. There is no A in a wobble position, and I don't know all the reasons for that. I wish I did, and probably somebody could figure that out pretty easily. G is sufficient, can interacts with both U and C, and U and C have enough ambiguity that they never code for 2 separate amino acids. Got all that? Me neither, but we'll go on. But anyway, you have to make sense. You have to make sense out of all of these data, and you have to make sense out of the chemistry of this loop if you want to understand origin of life. That's what I would say. This is sort of stating the problem. It's doing it in a long way, but I don't know how else to do it. So maybe more about this in a later webcast if you have the patience. So aminoacyl tRNA synthetase enzymes. So this is how they evolved, I'm going to say. And I would be the first one to say that what we did with this is not perfect, and we would like to improve it. But we need some bioinformatics support if we're going to do that. So these are all class 2 enzymes. Class 2 enzymes have an enzyme active site that's mounted on anti-parallel beta sheets. Class 1 enzymes have an active site at the C-terminal end of a set of parallel beta sheets. So 2 very different folds. But it turns out they're homologs. So GlyRS is very clearly a homolog of ValRS and ILURS if you look in the right organisms. To make these enzymes, you take this enzyme, you extend the N-terminus, and then you change the fold, and you get ValRS and ILURS. So I'm going to say that all the class 1 enzymes radiated from an ancient ValRS, and all of the class 2 enzymes radiated from GlyRS 2A. And here you have a bunch of 2A enzymes. The ones that are shaded in gray have separate editing active sites. That means if you have an tRNA alanine with the wrong amino acid, like serine attached to it, this enzyme will remove that amino acid. And that's in a separate editing active site. The ones that are shaded this color here have some kind of editing functions in their synthetic active site. The synthetic active site is the one that adds an amino acid to the tRNA. So basically GlyRS2A gives rise to all of these 2A enzymes, which are all closely related to one another. 2B enzymes, initially there was AsparS, and then asparagine RS came from that. Phi-RS is a 2C enzyme, and LRS is 2D. So this is like an approximate lineage through this set of enzymes. And over here, all of these 1A enzymes are closely related. These three have editing active sites. This one has lost its editing active site, although it has lots of sequence in common with these. I'm going to say that Val-RS is the root of this tree, but you could say that Gly-RS is ultimately the root of all of these enzymes, as I explained, because it gives rise to Val-RS and Ilu-RS. And then I'm calling ARG-RS and Cis-RS 1A enzymes. Other people would argue this, but this is a better characterization. They're part of this cluster. An ancient ARG-RS, I'm going to say, gave rise to a tyrosine RS and a tryptophan RS 1C, and probably also gave rise to a GLU-RS, Lyse-RS, and Glyn-RS, although this one has an interesting evolutionary history that I'll pick up subsequently. But basically, that's what you're looking at, and it's a lot to incorporate, and it becomes even worse on the next slide. Basically, these are all column 1 amino acids. So now I'm trying to correlate this map that I just showed you with the genetic code. So in column 1 of the genetic code, that's position 35 of tRNA, and it's A, 35A. So you have Ilu-RS, Lu-RS, Val-RS, and Met-RS. These are all related amino acids, and Val-RS appears to be the root of this lineage. Here in blue, you have Pro-RS. These are all 2A enzymes. They're closely related to Gly-RS 2A. This one has a separate editing active site. These have some editing features within their synthetic active site, the synthetic active site being the aminoacylating active site. And then you have His-RS2A. I'm going to say Asp-RS2A used to be, now we're in column 3. His-RS2A, Asp-RS2A, and asparagine 2S, RS2B. This one evolved by duplication of that one and then divergence. So these three are in green, and these three are shaded green: Lyse-RS, GLU-RS, and Glyn-RS. Glyn-RS is sort of like a modern eukaryotic innovation that's been distributed to prokaryotes, archaea, and bacteria by horizontal gene transfer. So I'm going to say that this pattern gives you back a lot of information about the patterning here. Here you have hydrophobic amino acids, 1A enzymes, that's evolution around the 35 base. You have red amino acids, serine and threonine are closely related. These are 2As. This one used to be a 2A and then changed to a 2D, got replaced in evolution to suppress translation errors. Orange, green, orange, green, orange, green. That's fractionation on the 34 base that I think I've covered up with our pretty mugs. And let's see. What else? And here you have 2-cyan. The amino acids are not closely related, but their charging enzymes are closely related, as I showed you. So there's a bunch of, if you're not bored yet, that's a bunch of future reading. Actually, those are all my papers, but I reference other people's stuff, I hope generously. And that's the story. So if you want to understand the origin of life on Earth, I hope I've convinced you that you need to integrate tRNA evolution, tRNAome evolution, aminoacyl tRNA synthetase evolution, and genetic code evolution. And if you can put all that together after just hearing this talk, then you're smarter than we are. It may require a little bit of study and homework on your own. But I think it's a cool story. It's just like the greatest story in 4 billion years. And I'm pleased and proud to present it to you. And if you're not positive of anything else, you should be positive about this. So this is the Rosetta Stone for the evolution of life on Earth. These are sequences, tRNA sequences have survived from pre-life, and they've survived so that the primordial sequences before living organisms on Earth are still clearly recognizable. So I think I'll stop there and stop sharing. And so where are we, Lei?
So there are several questions I'd like to discuss with you to help our audience understand this presentation. The first question is, why is it so important to understand tRNA evolution?
Right. So tRNA is a living fossil from pre-life, and it gives a sequence record of mini helix world and polymer world, which are both surprising because they have regular sequences in them. I wouldn't say that all the sequences and all the polymers in polymer world and mini helix world were ordered, but the ones that survived in tRNA are all highly ordered and regular sequences. So basically, transfer RNA sequences in living organisms are a history of pre-life on Earth, and they've survived for 4 billion years, and most things don't survive for 4 billion years. Some of us are a little bit more volatile at that.
The next question is, why is it important to understand the chemistry and dynamics of the anticodon loop?
Yeah, so the anticodon loop is really a specialized loop. It's really tight in the form it makes on the ribosome. And because it's tight, I think in the ancient world, it was protected from degradation by ribozymes that otherwise would have attacked it. It's got the U-turn, and then it projects 3 anticodon nucleotides, wobble, Watson, Crick, Watson, Crick. Basically, it's specifically chemically formed to support a 3 nucleotide code. And if you want to try and evolve a 4 nucleotide code, you'll never succeed. If you want to expand our 3 nucleotide code to a 4 nucleotide code, that's a big problem. People have tried to do that with limited success. Anyway, you have to evolve a 3 nucleotide code with 2 Watson-Crick positions and 1 wobble position. And if you can think of another way of doing it or another way of designing a genetic adapter that's as good or better than tRNA, go to it. I can't figure out how to solve that problem. That's a hard problem.
My next question is, why is it important to understand tRNAome radiation?
Right. So in this presentation, I didn't really show much about tRNAome radiation, but I did show aminoacyl tRNA synthetase radiation in 2 dimensions. I wish it was in 3 dimensions. Lei and I have struggled with this for a bit, and we're still trying to solve this problem. tRNAomes are the same. So if you built a lineage of tRNAomes, you'd like to see radiation from the primordial tRNA sequence to all of the tRNAs in an organism, and you'd like to see how all the nodes connect. And you'd love to see that for Pyrococcus and for other ancient archaea, because that would tell you a lot about how the genetic code evolved. We've published a paper on the subject, and I think it's pretty good, but it could be improved with better bioinformatics.
I have a question about the genetic code. So why is it important to make sense out of the evolution of the genetic code?
Yeah, well, so I don't think there's any evolution of life on Earth without a genetic code. So understanding the evolution of the genetic code is fundamental to solve the problem of why we have biological diversity. Until you can have RNA sequence dependent specification of protein sequence, you can't have biological diversity. You can't have biological complexity. You can't have anything that we would recognize as life without that kind of feature. So you need tRNA because you need to form a tRNAome, and you need aminoacyl tRNA synthetases because you have to link amino acid chemistry to nucleotide chemistry at a ratio of 3 nucleotides to 1 amino acid. Basically, you need all of these features and the genetic code, otherwise you can't have life. If you try to build life on Enceladus, you'll have the same problems. So.
Finally, what's your opinion of late wobbling in evolution of the genetic code?
Yeah, so this is like a weird idea, and it was come to computationally. The computation seems to be quite sophisticated. And by that I mean I don't understand it myself, but the idea of late wobbling is very strange. So I've presented wobbling as an evolved feature. That is to say that degeneracy in the genetic code is due to evolution of wobbling. So initially you have wobbling at tRNA 34 and 36. You suppress wobbling at 36, and then you keep wobbling at 34 if you try to suppress wobbling at 34 to have a richer genetic code that encodes more proteins. At this point, that would be a lethal condition, so the organism would die. So basically, you have to have 2 Watson-Crick and 1 wobble position. You probably can't do it another way, and you can't understand that unless you understand wobbling. So I'm going to say, for me, and I hope for Lei, that late wobbling is a strange idea and probably is wrong. So are we done with another one?
Yes, it's
Okay, so now I have to figure out how to work this thing. Oh yeah.